So you've heard about the genome, you've heard about the proteome, but have you heard about the serome? The serome is the totality of a single individual's secreted antibodies. What they bind to, what they react to. Your adaptive immune system is perhaps the most complicated system in your body. Over 10% of all the cells in your body are antibody-producing B cells. The adaptive immune system is primarily a random shape generation mechanism. And so the more unique individual cells that you have with their own unique pseudo-randomly generated antibody receptors, the more opportunities that they have for one of those antibodies to recognize any possible virus or bacteria that essentially the entirety of evolution on planet Earth can throw at us. But it's generally essentially a random process. And so even identical twins will have a completely different immunologic and seromic profile. So while the population of naive B cells is impacted by both our genetics and random chance, the way that an individual's serome actually arises is via immune experiences that they encounter over their lifetime. These can be vaccines, a virus, the foods you eat, the things that you're allergic to, the microbes that live in our gut. It's the totality of our recorded life experience here on Earth. Given the immense scale and complexity of the adaptive immune response, it's not surprising that sometimes this leads to mistakes and our immune system attacking our body's own natural proteins. All of us are walking around with very strong autoantibody responses to essentially random proteins in the human body. Every time your immune system reacts to a virus or a bacteria, there's a chance for part of the literal thing that makes up that virus or bacteria to very closely match a literal piece of the human body. And so when this occurs, we call that concept molecular mimicry. And a lot of the times when this occurs, nothing bad really happens. But sometimes these autoantibodies are against really critical proteins, like signaling molecules or immune cytokines. Or sometimes they're binding to the outside of a brain's neurons and accidentally training the immune system to kill it. And so for that reason, knowing when, where, and how different autoantibody profiles arise is really important. And so identical twins, or essentially inbred identical twin-like mice, will have very different seromes from each other. Even if you have the same genetics and you expose something to all the same stuff, on the autoantibody serome side, it's going to look very unique. Recent work was done showing that a particular protein of Epstein-Barr virus, in some people, a portion of that virus cross-reacts specifically with a piece of a human protein called gliocam, which is expressed on our neurons. And so it's possible that about 25% of the total multiple sclerosis MS population are having their disease induced specifically by an infection with Epstein-Barr virus, cross-reacting with a natural protein in their body that then is causing their permanent chronic neurologic damage. The opposite case can also be somewhat useful. A tumor looks profoundly different to your immune system, or at least it can, from the rest of your body. That's because the mutations that lead to the cancer cell becoming cancerous also induce unnatural protein shapes that our immune systems can recognize. And so one additional really interesting application of seromics is for discovering novel tumor antigens that you could use for, say, a cancer treatment or prevention vaccine. How do you measure the serome? Well, first, you need a sample from a patient that has antibodies in it. You need some sort of antigen of interest to compare those antibodies to, and then you need a method of detecting an interaction. However, if you're trying to discover novel causes of disease, which are associated with individual patient seromic profiles, you need an omics scale assay, something on the scale of the entire human genome. And so one way to do that would be to synthesize, purify, and print every single protein in the human body on a single glass slide. This is HuProt, the Human Proteome Microarray. It is the only protein microarray of its kind, which represents the majority of the proteins in the human body on a single glass slide. You take blood and you react it with the slide. You next come back with a detection antibody, which can be read on a laser microarray scanner. The intensity of the light will identify which unique autoantibodies are occurring in different patient populations. Even individuals that have the same autoimmune disease will still have exceptionally unique seromic profiles. However, what you will generally find is there is some unique autoantigen that they share. Where you start seeing things shared across a cohort of patients is typically where you start to see the things that might be associated with a causative mechanism of the disease. One of the main purposes of a tool like HuProt is it has full-length, 3D folded human proteins. These are really valuable for discovering whether or not relevant autoantibodies are occurring, which bind the actual folded structure of full human proteins, as well as target identification and other uses for discovering new types of protein-protein interactions and whether or not antibody drugs are hitting their intended targets. While the majority of an individual's autoantibody serome is generally thought to be harmless, there are some very clear and very profound cases where this is obviously impacting human health. The immune system can actually knock itself out, almost as if there was a genetic mutation. And what these drug-like autoantibodies do is they function as a phenocopy of these inborn errors of genetic immunity. In COVID-19, they've shown that for some people, why COVID-19 infection is dangerous at all is in part because of naturally occurring anti-interferon autoantibodies, which seem to impact and disrupt their natural immune response to COVID-19. And patients that have been given anti-PD-1, anti-CTLA-4. So these are the two cancer immunotherapies. And in some patients, you give them the drugs and it completely clears their tumor. Unfortunately, for a significant fraction, over half of the patients, they die anyways, and it's not really known why. Using our HuProt microarrays showed that the seromic profiles of these melanoma patients before they get cancer immunotherapy, they have a signature written in their autoantibodies, which seems to predict whether or not they will respond. It means there's something going on with the immune response prior to you giving the drug, which actually determines the outcome of this really important cancer immunotherapy. There's thought to be around 3,500 different surface proteins involved on the outside of human cells, and each and every single one of those is a potential case where perhaps an antibody to it could impact its function. If the immune system's allowed to knock out cytokines, what else might it be doing? Could it be knocking out cytokine receptors? Could it be binding and altering the function of GPCRs? These are all other things that the immune system might be unintentionally doing that interfere with our natural biologic processes and could contribute to disease. For certain things, 3D proteins are really important. In other cases, it's more valuable to have more fine detail, and that's enabled by PhIP-Seq, or phage-display immunoprecipitation-sequencing. We're able to take these small little viruses that infect bacteria, and you can teach them to put a small piece of a protein of your choice on their external surface. You could take the universe of known human viruses. You could take the entirety of the known human proteome. You could take the entirety of a research model, such as the mouse proteome. Our two most important autoantibody PhIP-Seq libraries, HuScan and MouseScan, are an entire reference human and mouse proteome cut into small overlapping pieces that represents as shorter peptides all the proteins in the human or mouse body. You react an aliquot of your library with an individual patient sample. Next, you capture the antibodies which are bound to individual bacteriophages from your PhIP-Seq library. Next, you barcode, pool, and sequence all the captured phages. As we've seen with multiple sclerosis, by using the combination of a viral PhIP-Seq assay in comparison with our HuProt human proteome library, that work was able to show that a small piece of viral protein identified by a PhIP-Seq assay caused an antibody which cross-reacted with a human protein identified by a three-dimensional antigen on a human protein microarray. While the field of seromics is still in its infancy, we already know that there are some very powerful things that we can only do with this data type. There are pharmaceutical companies working on trying to determine whether their drugs will fail or succeed based on pre-existing autoantibodies in their patient populations. Additionally, people are discovering as yet undiscovered causes of catastrophic chronic diseases like multiple sclerosis, lupus, and neurodegeneration. A deeper understanding of seromics, powered by new synthetic biology platforms, will unlock new information, enabling doctors and scientists to better treat human disease.